So we do the stimulation in both sides, trying to modulate the connection between these two areas, which synchronize, but we can also do non-synchronized stimulation. There are specific activations in areas related to default mode network.
The talk I'm going to give you today would be about neuroscience and how neuroscience could reshape the future of addiction medicine and psychiatry.
In terms of the neuroanatomy of these areas, I'd highly recommend you an online course that I put in YouTube. It's free of charge. You can find it on YouTube. It's called Fundamentals of Cognitive Neuroscience.
But in reality, the next step would be, can we make network-based interventions?
And then the idea is there any chance that we can develop interventions that help us to bring in back the executive control network?
So I will try to cover 4 major topics today in the limited amount of time that I have.
Is there any significant brain response to these small electrical activities that we target inside the brain?
There is a question how these two networks are interacting with each other, executive control network and default mode network. And now we are doing things that we call closed loop TES-fMRI.
And we realize that these top-down regulations are highly correlated with the level of activation that we have when we expose people to drug-related cues. I'm really interested to what we call transcranial electrical stimulation.
And we have recently developed some COVID-related posters in this series regarding stress and resilience and how you can still keep on your recovery, is how these connections and these changes in connections would be related to the behavioral and cognitive outcomes. Books are available online. You can find them even on Amazon, so they are available in different places, so you can find them, and there are different versions of these books available. Development of technology is another really complex issue, and we need to invest on that as well. And these are basically this is an overview about all these networks I talked about today: INTF, INTOM, ISAM-NIC, and Macri. So these are the different networks that have people.
Hello everyone, I'm really happy that I'm here, and thank you very much for the invitation. The talk I'm going to give you today would be about neuroscience and how neuroscience could reshape the future of addiction medicine and psychiatry. I'm from Laureate Institute for Brain Research in Tulsa, Oklahoma, and our center is basically founded to be able to reshape the future of psychiatry with different neuroscience technologies. That is the main mission in our center. And if you do not have a clear idea where are we located in US, we are located in the state of Oklahoma, which is one of the least kind of healthy states in US. And there are many problems and issues regarding substance use and mental health. And that is one of the main motivation for me to work in the field of psychiatry and mental health. And there are definitely lots of potentials for reaching out to the communities and trying to see how we can help people in different communities. So this is the place that we are located in the state of Oklahoma. And I do not have any conflict of interest. Let's just start with this kind of classic story from the classic neuropsychological text, that when we do neuropsychological assessments, we usually have patients with different lesions in different parts of their brain, and then we just try to see, okay, if they have same symptoms, we try to find, okay, what would be the common neuroanatomical substrate of that specific symptom, trying to refer that specific area to that specific symptom. That is the classic sort of paradigm in neuropsychology. But in reality, we see that there are patients that they have same symptoms, but at the end of the day, when you localize their kind of brain injuries, you can see that they have injuries in different parts of the brain, and there is no overlap. And the question is why they still have the same symptom. And now people are thinking about what we call network-based pathophysiology. Thinking about, okay, it seems that those people who have same symptoms, but different areas in the brain, they are parts of the same network. That is the reason that they have same symptoms. So this is called network-based pathophysiology, which people are trying to promote in the field of clinical neuropsychology. But in reality, the next step would be, can we make network-based interventions? And that is a really new topic that people try to see is there any chance that we can do specific interventions in networks and try to modulate those networks? And this is going to be the major topic for my talk today. So I will try to cover 4 major topics today in the limited amount of time that I have. I will try to remain on time to give you time to be able to ask some questions. So I will try to cover 4 major topics, basically giving you an idea about network-based interventions, and I will discuss about how we can optimize targeting for network-based interventions, and then how we can individualize interventions, and how we can do networking, and what could be the next steps. If you are interested in this field, how you can collaborate and how you can join the networks that we have made around the world. So that would be the major outline of the talk that I'm going to present. So it is something like 2 p.m. in the afternoon in South African time. So I was just going to ask you to just close your eyes for just 30 seconds and try to do nothing. Just close your eyes for 30 seconds and just try to do nothing. Okay, so if you do so, if I put you inside a scanner and see how your brain would get activated, it's quite interesting that your brain would not just shut down. You have specific activations in your brain when you are supposed to do nothing inside the scanner. And we call those activations and then the interaction between these areas as what we call default mode network, or DMN. And default mode network has several nodes, including a node in an area that we call ventromedial prefrontal cortex, and an area that we call posterior cingulate cortex, and few other areas in inferior parietal cortex. So there are some other nodes that contribute to what we call default mode network. And if we do some other activities, let me just ask you a few simple mathematical questions. And you have your chat space, so you can just report the numbers in the chat space. So just open your chat space, be ready. I'm just gonna give you some numbers, and then you are going to calculate some simple mathematical equations. Okay? Ready? Okay. 13 times 12. Okay, 156. 15 times 16, 240. 23 times 21. Okay, if I just go ahead and give you several of these equations, there will be lots of specific activations in your brain that most of them are related to a network that we call executive control network, or ECN, which includes an area we call dorsolateral prefrontal cortex and posterior parietal cortex. So these areas would be involved in what we call executive control network. Okay, and executive control network is close to what we saw in the first few slides that I showed you from group in Harvard, Michael Cox. The network would be including the dorsolateral prefrontal cortex area in the parietal cortex, and also in temporal cortex and parietal cortex. So this is the figure that we saw before together. And then there is a question how these two networks are interacting with each other, executive control network and default mode network. And it seems that there is a sort of a balance between these two networks. When you have higher activation in default mode network, you have lower activation in executive control network, and vice versa. So these two networks are counterbalancing each other in a way that they can just get activated and deactivate the others, or the other could get activated and just deactivate the other network. So that is the way that they make a balance together. And it seems that there is another network that is managing this interaction between these two networks, a network that we call saliency network, or SN, that includes insular cortex and dorsal anterior cingulate cortex. So up to now, I put forward something like 6 different neuroanatomical terms, and sometimes people would say, okay, they are really complex to understand. Those who are interested to learn about these types of terms and how different parts of the brain work, and learning more details in terms of the neuroanatomy of these areas, I'd highly recommend you an online course that I put in YouTube. It's free of charge. You can find it on YouTube. It's called Fundamentals of Cognitive Neuroscience, and we are already in the session 12. And I have students from South Africa that Tanya introduced them to the course. So, and there are people, something like more than 100 students right now attending the course, but the offline videos are also available. So we discuss about those types of neuroanatomical key terms, and if you are interested, you can just take a look in this online course that you can easily find on YouTube. But the question is how we might be able to do network-based interventions, and how network-based intervention is going to be important for different psychiatric disorders. And I'm going to be mainly focused to substance use disorder, to see how we might be able to develop network-based interventions for substance use disorder. That is the question I'm trying to address today. When we want to talk about substance use disorder, we know it's a really prevalent problem around the world, and also in US. In US, from every 12 adults, we have one involved in substance use disorder during last year. So it would be something like 20 million people involved in substance use disorder. And if you want to include alcohol, then the numbers would go something like over 50 million people are suffering from a sort of alcohol use disorder problem. So we know that is a prevalent problem around the world. And to be honest, we do not have very good intervention. So it's just, there are some available options, but we still, especially for things like methamphetamine use disorder or cocaine use disorder, we actually do not have any intervention that would pass high quality standards in randomized clinical trial, double-blind randomized clinical trial. So there are challenges in this field that we know. And to have a better understanding about when we talk about substance use disorder, what are we talking about? Just let me give you a few examples. If I show you a chocolate cake, and if I want to ask you just think about the best chocolate cake you have ever had in your entire life, and think about when you see a chocolate cake, what would be the cognitive processes that get involved in seeing this chocolate cake, and just try to compare the cognitive processes or the brain processes that are involved when you see just a box of napkin compared to seeing a really fancy cheesecake. And you know that the main thing that is involved is what we call craving, or what we call cue-induced craving. So that is a really important fundamental brain function that includes different kind of sub constructs that are involved in what we call cue-induced craving. And it is the case not only for, food and things that are related to our survival. Among those who are substance users, they have cue-induced craving related to anything that is associated with substance use. And that is one of the main reasons that these people would come back to use the drugs again and basically relapse to substance use. And cue-induced craving, or craving in general, is one of the major challenges in the field of substance use disorder. And the question is how we can just understand craving, and then how we can do interventions for drug craving. And what we do, we put people inside the scanner, we show them neutral pictures and drug-related pictures, with the hope that we would be able to find the neural circuits and networks that are involved in substance use disorder, and then thinking about how we can develop interventions for modulating drug craving. And this is the way that we do. We put people inside the scanner, show them neutral pictures and drug-related pictures, and just make the contrast to find the networks that are involved. And when we put these people inside the scanner, what we can see that there are specific activations in areas related to default mode network when we have cue-induced craving. And there would be less activations or kind of less engagement in areas that are related to executive control. So that balance would go for default mode network and cue-induced craving when we expose people to drug-related cues. There are some minor details and challenges. I'm not going to go to the details, but an overview is probably we have a balance towards the default mode network when we expose people to drug-related cues. And then the idea is there any chance that we can develop interventions that help us to bring in back the executive control network using saliency network to be able to give people better level of control to control the drug craving. So that is the idea for network-based interventions. And we have different what we call non-invasive brain stimulation technologies developed during last 2 decades. That people are using these technologies to be able to target some nodes in those networks, trying to see if we can modulate those networks. And what I'm more interested to among these different sort of technologies that we use in our lab, I'm really interested to what we call transcranial electrical stimulation. Why I'm interested to this technology? Because it is inexpensive. It is you can buy the devices with a couple of $100s, and the technology could be really scalable. It's going to be really handy, and you can use it in different situations. The challenge is there any effect for these sort of transcranial electrical stimulation with just a little bit of current that could be produced by a small 9-volt battery? So that is the main question. Is there any significant brain response to these small electrical activities that we target inside the brain? We have done studies, for example, among methamphetamine users. We have done this study something like 6-7 years ago, and we showed that if we put electrodes over prefrontal cortical areas, we can modulate drug craving over the time. So we can reduce the level of drug craving among substance users. And that is what we have found. And it has been replicated by many other colleagues around the world in different labs. And people have a sort of conclusion that there might be potentials that we can modulate drug craving, as we have discussed, with these brain stimulation interventions. But the reality is what we have in terms of the changes in the level of craving before TDCS and after transcranial direct current stimulation. There is a huge variation in terms of the response. People have different types of responses. And the question is why? What is really happening? And to be able to understand what is really happening, we have done a study with fMRI. So doing fMRI and imaging from the brain before and after stimulation to see if we can see the changes inside the brain with this sort of a stimulation. And the stimulation was the same as we have done before, with putting the anodal electrode on F4 and cathodal electrode on F3. And the idea was, okay, is there any chance that we can modulate the interaction between default mode, executive control, and saliency network? What we saw was basically, yes, it seems we can. It seems that there are pieces of evidence that we can modulate the network connectivity from having default mode network engaged to have executive control and saliency network engaged in this process. And we have published the results, so you can take a look and so. So it seems that we might be able to modulate these larger scale networks with just stimulating prefrontal cortex, that there are hopes for that network-based intervention. And it is not the case only for substance use disorder. It seems that other psychiatric disorders could also benefit from these sort of network-based interventions. Think about depression. This is a nice review paper by Emmett Etkin from Stanford, that they have suggested that probably modulating dorsolateral prefrontal cortex would help us to modulate executive control network. As you can see here, the frontoparietal network is the executive control network. And then the saliency network and default mode network that are connected would be influenced by that frontoparietal network modulation. So that is the idea that people are following, not only in substance use disorder, but in all the other psychiatric disorders as well. So in the same context, we have started to think about, okay, if we are thinking about executive control network as dorsolateral prefrontal cortex and posterior parietal cortex, should we do a stimulation on both sides and engaging the network in both sides? That is another idea that we have recently started a trial with, what we call frontoparietal synchronization. So we do the stimulation in both sides, trying to modulate the connection between these two areas, which synchronize, but we can also do non-synchronized stimulation. But in this trial, we are doing synchronized stimulation to see if synchronized stimulation could help to modulate drug craving. And we are in the something like the middle of the trial. But in terms of being able to find the best target for this stimulation, we started thinking about, okay, if we do the stimulation on frontal areas and then parietal areas that I showed you, the frontoparietal network, and the area that we called F3 and F4, we are targeting F4 right now, and P3 and P4, we are targeting F4, P4. So these are the sites that we are targeting right now. How these areas are interacting with each other. So we have done a frontoparietal connectivity during drug cue reactivity, and we didn't find any connection between F4, P4, and F3, P3 during drug cue reactivity. And that was a pretty much bad frustration, because we have been targeting this network and we really like to see this network is involved in drug cue reactivity, but it was not. And that was a sort of headache for a while, thinking about, okay, why we have this issue in terms of not having the frontoparietal network engaged in drug cue reactivity when we measure this connection. The frontal areas are a little bit connected, I mean, negatively connected, but no connection between F4 and P4 and F3 and P4. And we have done that with kind of in different ways, but the connection is not there. So what we have done a whole brain analysis from those sites in frontal areas, in right and in left. As you can see here, when we do a whole brain analysis, we have the connection with the node in parietal cortex. And if I do it the same again, and from the right side, in the whole brain analysis, there is a site in parietal cortex. But obviously, this site is not what we have been looking to in the parietal cortex. So we put those activations over the cortex, and we try to see, okay, if this is the area that we are interested to, let's see how these areas are related to the electrodes and positions of electrodes over the skull. And realize it is not basically P3, P4, but it is CP5 and CP6 networks or nodes that are involved in this network. So we change the target to these nodes, and now these nodes are connected to each other. So we realize that if we change that from P3, P4 to CP5, CP6, we can find the areas that are connected. So the network is not actually located on the top, on the upper side, but is a little bit lower and a little bit more towards the anterior side. We have done the same with resting state connectivity with F3, P3, and you can see that these areas are even negatively connected in resting state. But if we do the same site that I have mentioned, they are highly connected in resting state fMRI as well. So that is what we found in terms of, it seems that these areas are in the same network, and then we can target them. But going back to the idea of, okay, if we are talking about frontoparietal areas, why when we are targeting, let's say, the frontal areas, we do not have connection with the parietal area? We realize that we have a site here. This is what we call UO-atlas. In UO-atlas, which is a network-based atlas, there is an area in the frontal area, as you can see here, which is in purple color, as you can see here, which is also connected to the areas here. And this network is named ventral attentional network. And ventral attentional network is another name for what we call saliency network. So it seems that the site that we have in the frontal area is not actually a part of what we call executive control network, which is the orange one. This one is the saliency network. And the saliency network, as I told you, would involve insular cortex and also dorsal anterior cingulate cortex. So we have a certain thing, but okay, it seems that we are tapping into another network, not the executive control network. We are tapping into the saliency network. So we start to think that, okay, if it is the case, then we should have connection to insular cortex, because insular cortex is a part of the saliency network. And we checked that with both task-based drug cue reactivity fMRI and resting state, and we saw, yes, it is the case. I mean, these areas are connected, especially with the anterior insular cortex. They are connected. And also with anterior cingulate cortex, these areas are also connected to anterior cingulate cortex, especially the area that I told you, dorsal anterior cingulate cortex. So it seems that dorsal anterior cingulate cortex and anterior insular cortex, they are involved in this connection. So it seems that we are tapping into this network, not the network that we have been thinking about before. And it seems that this network is also connected with subcortical areas like nucleus accumbens, amygdala, and ventral caudate. And it seems that there is something like a top-down regulation with these areas. And we realize that these top-down regulations are highly correlated with the level of activation that we have when we expose people to drug-related cues. So it seems that the higher connection, the higher the lower the level of the craving that we had, the activation that we have, or the activations in the craving would be related to significantly to the level of, let's say, top-down connections that we have in this network. So it's just getting more and more interesting in terms of, okay, how we can explore more this specific network. So we have a certain thing, but okay, we had a kind of a database with fMRI data. So let's go back to this database and see if we have top-down regulation, as we discussed about today, between these frontal areas and subcortical areas that we have been discussing about. So we brought again the database that we had, and we realized, yes, there is, when we just consider the node under anodal electrode here, which is a node that we were stimulating with TDCS, there is a significant higher connection with anterior insular cortex here, as you can see here on the right side, so right to right. And then we have a negative relationship with dorsal anterior cingulate cortex, as you can see here. And if I do that with left cathodal, which we are inhibiting or suppressing with cathodal stimulation, we have negative relationship with anterior insular cortex in both sides. So it seems that, yes, it is the case that we are tapping into the saliency network, and we are modulating insular cortex and anterior cingulate cortex, and that is basically
probably the network that we are tapping into and we are modulating. So it seems that this is the case in terms of we can modulate these specific networks and target them. But just going to the second part of the issue in terms of, as we have been discussing, how we can optimize. And as you realize, it is not easy as just saying that, okay, this is the frontal area, this is the parietal area, we just stimulate and we can find the modulation effect. We should be really careful that when we talk about these networks, they are highly close to each other. And you can have multiple different networks in frontal areas and multiple different networks in parietal area. And optimizing the targeting and clearly knowing that where you are targeting is going to be critically important for you to be able to have the desired result that you are looking for. And even those who have been thinking about, okay, we can just do dorsolateral prefrontal cortex stimulation and parietal stimulation, and these areas are connected to each other. They know they need to be more careful in terms of, okay, when we talk about prefrontal cortex, we have areas related to saliency network here. We have areas related to executive control network here. We have even areas related to default mode network represented in prefrontal cortical areas, and some other networks, as you can see here. And we need to be careful that when we talk about these networks, it might be another network that we are discussing about. So we need to clearly define how we can optimize the network intervention. And targeting optimization is going to be a big challenge and important issue if you want to go for this area of research. The third issue I'm going to cover a little bit today would be about individualization. And why do we have these nice walnuts here? Anybody has an idea? You can use your chat space. I'm not sure if the chat space is available here, but yeah, you can see that people are okay. Why we are using what we call these beautiful walnuts? Obviously, we know that people are different. The brains are quite different from each other. How we can do individualization? How we can really find, okay, if this is the case for one case, do we have the exact same network structure or different network structure? So we need to optimize the stimulations per individual. That is what we definitely need to do in the future. And to be able to do that, we need to use brain imaging. We need to clearly define what are the networks and how these networks are working. And now we have started to work with watermelon. And why watermelons? Because they are really good for stimulation. So we put the electrodes over watermelons and try to see how we can, is there a chance that we can do a stimulation while we are scanning inside the scanner? So that was the question. And so we put the caps over watermelons and we made a setup in our lab being able to do that. And we started to do the stimulation and recording in watermelon to see if we can do the stimulation. Because it's really complex to do the stimulation inside the scan. There are lots of noise, and it's critical to consider lots of technical details to be able to do that. But it seems that we have been successful. As you can see here, we can easily see inside the watermelon what is happening inside watermelon, and we can do the stimulation as well. And of course, when we do the stimulation, after the stimulation, watermelons, taste really good. So we just, we always eat the watermelons afterwards. And the next subject after watermelon would definitely be my brain. The students love to stimulate their PI brain. So I was the next subject going inside the scanner and trying to see how we can do that on human brain. And it seems that we have done different pilot studies. It seems that it is going to work perfectly well. So we designed different electrode montages, different types of electrodes to be able to do the stimulation inside the scanner. And some of these electrodes are the first time that people are using this size of electrodes that could be, that make it possible for us to stimulate two different sites in two different kind of combinations to be able to do network-based intervention inside the scanner. And this is the setting that we have right now, being able to do the stimulation inside the scanner. And we have a postdoc, Alki, and a PhD student, Benny. They are working on that specific project. Brilliant students, and they are doing a really nice job for this. And this is the way that we try to record the connection and network activities and also stimulate. So that is the way that we put all these things together. And now we are doing things that we call closed loop TES fMRI. So we record, we compare the connections that we have with the target that we have in terms of we really like them to get connected to each other. We optimize the stimulation protocol in terms of the frequency of a stimulation, the phase difference between the stimulation sites, and we record again. And that is the way that we do what we call closed loop TES fMRI, to be able to find the best frequency and the best target and how to engage those networks in the brain to be able to engage them in the process that we are interested to. We have some pilot data, just kind of the really initial steps that we are doing, but it seems that this is not an experimental group or control group. This is one single subject, basically two single subjects from the experimental group and control subject. And you can see that the frontoparietal connectivity was higher in the experimental group. And this is how we search for different phases and frequencies, and how the optimization protocol is working, and how we can find the optimized stimulation, and how not only the connection, but also the behavioral responses in terms of the accuracy would go higher for experimental group, but not for the control group. So we try to see how we can modulate these connections inside the scanner. And it seems that there are, we are still in the really initial steps, but it seems that there are hopes for that. And in this idea, I'm just going to give you a better sense about how the system is working. So we are targeting, let's say, executive control network and saliency network as our neural target for of interest. And then we have, as I told you, we have been doing the interventions and considering frontoparietal connectivity as the what we call neural target engagement measure, and we try to improve that. So that is the way that we design this sort of network-based interventions. But the next question that we are still dealing with that is how these connections and these changes in connections would be related to the behavioral and cognitive outcomes. And finally, how they are related to clinical outcomes. Do they have any meaningful clinical outcome if we improve connection between DLPFC and posteroparietal cortex? We still do not know. We are still exploring, and we are still seeing how we might be able to find a specific clinical benefit for that. But we probably need another, let's say, 5 years to be able to be back to give you a better sense about is there any clinical effect or not. But we are still exploring about potentials and how this system is going to work in the next steps. Okay, so that was an overview about what we call network-based interventions for substance use disorder. But the question would be, okay, if you're interested, if you feel that it is an interesting field for science and you are interested to get engaged, I'm going to give you a few slides in the last part of my talk about potentials, how you might be able to get involved in the process that we are doing, which are called networking in network-based intervention. First of all, it's quite amazing to see that, for example, for doing brain stimulation in the field of addiction medicine, we have labs around the world from Netherlands, Israel, Iran, Czech Republic, Canada, Brazil. So there are different countries that are contributing to these brain stimulation studies in different clinical population. And it's amazing. It's amazing that people around the world are working in these activities. We realize that these labs are totally separated from each other. They are doing their own job. There is no common interaction between these labs. So we decided to make an international network of TS/TMS trials for addiction medicine. We called INTOM, and we have started to put lots of activities together for INTOM. You can just search for INTOM in YouTube channels, and you can find webinars that we have done and discussions that we had over the last couple of months about how these collaborations could be happening. We put together a consensus paper with 64 authors around the world, different labs. What would be the roadmap and the road ahead of us if you want to do kind of joint collaboration on brain stimulation for addiction medicine. And then we also made another network of those who are interested to do imaging and transcranial electrical stimulation. We call it INTF. And for INTF, we already have 54 labs in different countries around the world collaborating together to develop this technology, because development of technology is another really complex issue, and we need to invest on that as well. And then we also realized that we need to improve the fMRI imaging measures that we are using for drug addiction. So we made another network we called fMRI Drug Reactivity Consortium, or within what we call Enigma Consortium, which is a really large neuroimaging consortium around the world. So we made this Enigma ACRI consortium together. And I think that one of my colleagues, Panyan, is right now online. So Panyan, if you have access to the chat space, you can probably post some of the links related to these networks and papers, so people can start to find those activities around. That would be helpful as well. Okay, this is the list of the people who are involved already in Enigma ACRI consortium, and we are still accepting new members around the world if you are interested to join us. If you have any plan to do imaging and fMRI imaging in the field of substance use disorder, you are highly welcome to join us in this collaborative network of sharing information and expertise and protocols for the field of brain imaging for developing treatment for substance use disorder. And the last network that I'm gonna give you an introduction to would be a network that we have in the International Society for Addiction Medicine. We have a neuroscience interest group, which is mainly focused to any aspect of neuroscience that will be related to addiction medicine. And we have 200 members, more than 200 members around the world. And Panyan, who's online right now with me, is the kind of executive secretary of the network. And we highly encourage those who are interested in this field to join us. We have put together a special issue in Frontiers that discuss about brain and cognition for addiction medicine from prevention to recovery. When I put together these slides and some of these slides, we had something like 40,000 views. Few weeks ago, it was 87,000. And I checked it again last night, and it was 100,000 views in the specific special issue. So it seems that people are getting more and more interested to this area. And we put together in that special issue a paper we call a roadmap, how to integrate neuroscience to addiction treatment. And that is how we want to move forward with these ideas of neuroscience in addiction medicine. The other thing, that the last thing I'm going to target today, is when we talk about networking, we cannot just talk about neuroscientists. We need to communicate with public audience. We need to talk with people in the communities. We need to talk with our patients. We need to communicate whatever we are discussing about in terms of neuroscience and brain and all the other things to the patients as well. So we need to give them an idea, what are we going to do? What are we going to use for different sort of interventions? So we started to develop different sort of what we call neuroscience-informed psychoeducational materials. And we are running different studies using them for recovery. And also we have recently started to use them for prevention as well. And these materials, I've just provided you with some of these posters talking about how brain is involved in drug addiction and how you might be able to help your brain in the process of recovery. And we have them in 14 different languages. I'm really interested to see if anybody from South Africa would be interested to translate them to some local languages in Africa. And we definitely would be happy to help for that if you are interested to have those posters. And we have both a male version and a female version for these posters. So we realize that we need to consider all those gender issues as well. And we have recently developed some COVID-related posters in this series regarding stress and resilience and how you can still keep on your recovery while the situation is really complex. At least here in US, things are really messy. So we hope that the people can use these educational materials to help them in the process of remaining in recovery during the pandemic and the situation that we have. And these are some of the pieces that we have discussing about attention, how attention is involved in the problems that we have with COVID, and how we might be able to modulate attentional activities while people are in the quarantine or pandemic. These books are available online. You can find them even on Amazon. So they are available in different places. So you can find them, and there are different versions of these books available. And these are basically this is an overview about all these networks I talked about today: INTF, INTOM, ISAM, NIC, and IGMACRI. So these are the different networks that have people contributing to these networks, and we have colleagues from South Africa that are involved as well. So I'm hopeful that people would be interested to join these networks and collaborate with what we discussed today about basics of neuroscience, informed network-based interventions, how we can do optimized targeting, individualization. So these are the topics that we discussed about today, and just few take-home notes. First of all, I believe that network-based neuroscience and network-based interventions would definitely have lots of potential for future. And you can definitely learn about lots of really good potentials, how we can get these networks involved. And this is not just about brain stimulation. Even with psychotherapy and different sort of cognitive interventions, we know that how we might be able to change the interaction between networks using these different sort of psychotherapy or even pharmacotherapy interventions. So there are potentials for getting these networks engaged in this process. And also, as we discussed about, there are lots of networks that could help us in the world of neuroscience to be able to share our protocols, collaborate, and increase the quality of some of the studies. And that is what I'm really hopeful that we need to invest on in the future to be able to successful in the field. And at the last point, I highly recommend you to consider kind of communicating with public audience, and that is going to be an important part of this process if you want to be successful in long term. I should thank my colleagues, collaborators. There are many people who have been contributing to the slides that I showed you today. Different students like Ghazale, who was involved in some of the analysis that we have discussed about, many other colleagues and collaborators in different labs around the world. And I hope that to have your name in this list in the future as well. Okay, thank you very much for your time.